Welcome to lesson 1 of the soil restoration course. Our first lesson is going to be on soil formation and composition. So we'll look at how soils are actually formed, the processes involved, and what soils are composed of. So let's introduce the concept of soil formation. How is it made? So today we'll discuss how soil is formed from bedrock, what is soil texture, and that's the sand, silt, and clay component. And we won't discuss this in detail, but it'll give you a good grasp of the matrix that soil has, which is the mineral component. And we need to understand that it's a living thing, so it's organic matter and the microbes together with the sand, silt, and clay that really create soil, that what we can actually call soil. And organic matter is a huge component of that, so we need to acknowledge organic matter and understand what that actually is, its importance in the actual soil context. And soil microbes and the soil food web are what converts this organic matter and the minerals into plant food. So how these microbes and how their particular roles in a soil food web actually produce soil. We'll also talk lastly about the ecological succession of how the microbial diversity in soil drives above ground ecological succession. So the different plants that grow towards more and more biodiversity. I just want to draw your attention here to this beautiful diagram here to show you the very thing that we'll be talking about in terms of the different life forms, the different variety, the different interactions that the organisms have that actually make soil what it is, because the sand, silt, and clay component of soil is literally just the matrix which they use and the food from which they gain minerals from in order to feed themselves and the plants. But without all of these organisms being present and cycling nutrients throughout the soil food web, soil is not a possibility. Soil does not happen. It will just be dust that flows off into the atmosphere. So it is microbes that make organic acids. So fungi, for example, make organic acids, and alkaline compounds produced by bacteria, and the enzymes also being alkaline and acidic, that actually break down the crystalline structure of rocks into sand, silt, and clay. And here is an example of collaboration between organisms and their ability to decompose rock, because lichens are basically symbiotic relationships between variety of fungi, including yeasts, and variety of bacteria, and algae and cyanobacteria, and all of that plays an essential role in covering bare soils, such as soil crusts often contain lichen in them or must contain lichens in them. But also you can see them grow on trees and doing their job there as well, gaining minerals and photosynthesizing. And that's the beauty of it, they actually photosynthesize as well. So lichens are these strange cooperative little communities of organisms doing their job, and because the fungi and the bacteria are able to gain minerals from a rock and pass that on to the algae, and the algae photosynthesize, that's like a mini ecosystem that then translates into larger and larger plants. So if you can understand this concept of collaboration on this scale, we can also then translate that into how plants live and cover the mass of land that they should cover, that we live on. What's really important is to understand that soil actually is also created from below ground. So where we get to the parent rock material, that actually is being decomposed as we speak. And scientists have never really been able to show that until in 2019, a PhD student has shown that when you go all the way down to the bedrock material and basically gain that material and expose the bedrock to whatever is surrounding it, basically the idea is that bacteria can actually degrade that, and that's never really been shown before, even though scientists have always known that it must be happening. So these organisms are chemolithotrophic ferrous iron oxidizing bacteria. Chemo just means chemical and lithotrophic means living off rock. And basically they degrade the subsurface ferrous iron and gain their nutrients from that, while they decompose this iron silicate minerals in quartz diorite. So basically the parent rock was ground up, so cores were drilled, material was picked up, for the purpose of speeding up the experiment rather than waiting potentially hundreds of years for this to happen and to demonstrate it, they basically ground up this parent rock material and inoculated it with the drill hole material that surrounded that, and left it for 2.5 years, and then periodically checked what was happening. And there was a radical change in the surface texture and accumulation of nanoparticulate iron oxyhydroxides occurred when the bacteria were present, which did not occur where the bacteria weren't present. So this is what happens, how the rock got degraded here, being oxidized, and this is its original structure when no organisms are present. So basically it is a chemical and physical cracking phenomena that occurs first. This allows bacterial entry, and extracellular electron transfer happens where the iron is oxidized and coupled to adenosine triphosphate, which is the energy molecule of all living things, is generated. So that means energy is generated through this process for the microbes, and iron is the primary energy source for the growth of these organisms. So this is basically, in a nutshell, how it's microbial or biological degradation of rock, and not just cracking and weathering. Yes, some of these processes occur where water does have some impact on rocks, obviously, and removes the minerals, and there's obviously physiochemical processes, but it is the microbial processes that are of interest because they're the least understood, as well as hugely powerful, as you'll see soon when we'll talk about bacteria. But it is a community effort, and there are things that the bacteria produce, and these are called biofilms, where they grow on the rock surface, producing these mucilaginous polysaccharides, extracellular polysaccharide matrix, sort of hydrated gels, and they protect the microorganisms and provide adhesion, but also their communication sites. And that's where there's concentration of organic acids and siderophores, which are iron chelating or iron grabbing molecules, and weathering agents. So in these biofilms, the community of bacteria get together and actually produce these substances that allow them to live off the rock and therefore extract the minerals from the rock. And also this substance favors cell-to-cell communication and microbial interactions. So it's like their home. They're adhering to the rock. They produce all kinds of substances while they're consuming the rock, and making minerals present in their body as a result within their little communities, within their biofilms that they produce. So in that case, the way soil is really formed from below and from above, as the bacteria decompose this bedrock material to start with, parent rock is then formed, and then subsoil is formed, slowly being degraded. But from the top, of course, we've got plants growing and microbial life happening and all other processes, and moisture coming in, and air, and organisms moving in and throughout the soil, and basically creating that humic layer, organic layer, and that travels down with roots, organisms, and moisture through to the topsoil. And then also the alluviation layer is where leached minerals and organic matter is deposited further down the soil as well, creating the subsoil. And of course this is working hand in hand from below and from above. So this is the same sort of idea, but basically that's the layers that we see in soils, and they're created from both below and above biologically. So let's just look at the next image to show you some of these examples of these beautiful soil profiles. And you can see that those layers clearly here. We've got the rock parent material and all these layers, and the organic matter is where obviously that supports the growth of vegetation above. And yeah, just beautiful sort of a mass scale view, and then something that you can look at with your own eyes if you make your own soil profile examination. So quite a beautiful process. You can see all this stuff happening throughout the layers, and you can then understand that when you start with rock, you will really pretty much not have much growing, because it is those layers that are built up the top that allow plants to grow. So if we're trying to grow a forest on rock material, it's obviously not going to happen. But to go back to the mineral part of the soil, we have this test that you can do, and basically put a bit of your soil in a jar and see how much sand, silt and clay you will gain from that, or what is actually happening. And so it's a very simple way of understanding what sort of soil you have in terms of the mineral component that's there. And you can even look at it via the soil texture triangle. It's a pretty simple system. You just go and look at the different percentages that you got, and basically where they intersect, this is where your soil forms into. Here we go, 8, 29, and 63. So at 8% clay, 29 silt, and 63 sand. If you go from those edges and where those lines intersect at those percentages, that will be where your soil falls into. And sand and silt particles create air gaps and allow rapid water infiltration. They find it difficult to hold moisture and grow plants, and we need organic matter in them in order to actually sustain some water and life in them, whereas clays pack together tightly and have slow infiltration and produce waterlogging if they don't have organic matter in them as well, because their particles are so tiny. So if you have about same amount of 35% silt, sand and clay, if that's your ratio, you actually end up with a loam, which is the preferred soil type most people want to grow stuff in, because they avoid those problems of infiltration, soil aeration and waterlogging. But microbes and organic matter can change clay into loamy texture, and the same with sand. So that means that if we just keep encouraging microbial growth in the soil by adding organic matter through plant debris, we can actually change soil texture to resemble something that the mineral component itself is lacking. And just to explain that a little bit, you can see the different sizes of clay particles versus the larger particles like silt and sand. This is sort of gravel, sand is bigger, and clay is too tiny to even look at this millimeter scale. And so that's kind of what they look like. And the size and mineralogy of soil is looked upon as something that's a way to understand what sort of soil you have. And we know that silts are larger than clay, and then clay particles are classed as less than 0.004 millimeters, which actually is 4 micrometers. But geologists and soil scientists define clay as being 2 micrometers in diameter, whereas sedimentologists at 4 to 5 micrometers in diameter, and colloid chemists 1 micrometer. So there is no consistency in how clay particles are defined. And then geotechnical engineers just look at the plasticity characteristics of the soil. So really it is just in the eyes of the beholder as to how we classify clay, but it's really not that important. To what we want to achieve is to make soils filled with life again, because the organisms will deal with those particles as they exist. It is worth mentioning clay properties, as clay is the most studied component of soils normally, and it does have an effect on nutrient exchange within the soil and the type of minerals that are bound to the clay particles. They have this capacity to hold water and dissolved plant nutrients, and therefore are important. And also they give the soil its chemical and physical properties, such as swelling and shrinking and cation exchange capacity, which we'll talk about in a minute. Clays are very structured. The structure is based on the basic crystalline units of clay minerals, and there are 2 major types, and that's aluminium oxygen octahedrons, which are composed of oxygens and hydroxyls in the octahedral structure of aluminium, iron, or magnesium. There are also silicon oxygen tetrahedrons, which are silica sheets with 4 oxygen atoms. The tetrahedral sheets are bonded to the octahedral sheets of small cations such as aluminium and magnesium, which are coordinated by 6 oxygen atoms. So this is actually quite complex, and you can see on the image on the right that the different clay mineral particles that have different names that you might have heard of, like elite and smectite, they then form these clay mineral layers, and then the actual mineral sheets that are either tetrahedral or octahedral, and then that octahedral structure also changes depending on the kinds of minerals that are present and how they are arranged and how many sites they take up. So this is quite a complex science, and there's huge amounts of research done on this, and I won't go into it because I have no idea about it really, and also it's beyond the scope of this course. But if you wanted to have a look at that, there is lots of research on it. However, what's really important with clays is that they have this capacity to exchange cations. So it's called the cation exchange capacity. And basically, this depends on the crystalline structure and the charge that's on the clay, which then determines absorption or retention of cations. And these are the positively charged ions. So the definition of cation exchange capacity is the total amount of cations measured as milliequivalents that are held by the soil components, particularly the clay component of soil, and which can be exchanged with the cations in the soil solution. And you can see here that this negatively charged clay or organic colloid, which also has this capacity, so organic matter is very important if you don't have clay in soil, because it can serve a similar function. And you can have these positive cations attached. However, all of those positive cations can be exchanged with other positive cations and are interchangeable, so they can actually replace each other and will either be in the solution or be attached to the clay. And this basically drives sort of soil nutrition in a way, in a sense that this cation exchange capacity determines what plant available minerals are present in the soil solution at any given time. And the more clay and organic matter there is in the soil, the more positions are present to hold cations, and vice versa. The lower the clay content, so the more sandy the soil is, fewer positions are present to hold cations, and therefore sand has lower cation exchange capacity compared to clay. So this is basically how it's all measured. And this cation exchange capacity depends on the clay content, so the soil texture, the type of clay, so that depends on the mineral structure of the clay, and the organic matter content in that soil or in that clay will also influence that cation exchange capacity. And so therefore this is how soil nutrients and fertility are normally measured, by this exchange of cations either being bound to soil particles or being in solution and therefore available to plants. But what I want to drive here is that this whole idea of having nutrients in the soil, and some soils being devoid of nutrients and some having more than others, while it's true, there's actually no soil on this entire earth that is actually deficient in any minerals. And the reason for that is because the parent rock material always has minerals in it, all of the minerals. And there's a book actually written, it's called Environmental Soil Chemistry, and it's at 2nd edition currently. And so basically what's happened is scientists have gone around and tested every single soil they could find, and sediments and Earth's crust, and basically found that all of the minerals are present. None of the soils ever have a zero of a particular mineral. The only problem is sometimes we can't even see them just in the soil by doing a soil test. That doesn't mean they're not there, they're just not being detected. And the reason for it most of the time is because there's no microbes taking that away from the sand, silt and clay. That mineral is just bound up so much. But when you look at the actual averages in the soils, while some of the minerals are naturally low, remember that plants and animals only need tiny little amounts of these. But as long as there's some organism that's able to detach them from the soil matrix and put them into solution for the plant, we will never be deficient in those minerals. So this is why forests never need minerals, because they're getting them from the microbes, and they actually have very low mineral content in them, the soil themselves I mean, but the minerals are definitely bound up in all of the giant vegetation that's growing them and in all the animals. And so just a big there is no soil deficiency possible. It's impossible, because the parent rock material has every single mineral that Earth can ever have. So when you look at that reality versus what the ag industry false marketing says so that they can keep selling phosphate and nitrogen and other fertilizers, when we look at the actual amount, for example, of nitrogen and phosphorus, and we do a little calculation where there's average 2,000 kilograms per hectare of nitrogen, which is in milligrams per kilogram or parts per million, if you like. That's way over the crop requirements, because corn, for example, needs only 200 parts per million. So if you calculate that out, it gives you about 10 to 20 years in a single gram of soil to grow corn. And that's our nitrogen. And then phosphorus is a similar calculation, where phosphate fertilizer is running out, so it's a problem. And we're actually mining very delicate habitats, destroying whole islands just to get this guano, which is a bird poo that has been accumulated, and we're using that to fuel our food supply, but that's going to run out, and it's already happening. And most of these nutrients never actually get absorbed. They just go into waterways and kill our oceans and cause all kinds of problems. But an average soil will have about 800 kilograms per hectare, and 8 to 10 micrograms per plant per year is actually required. So that gives us about 100,000 years worth of phosphate just in your soil already. So why would you buy it? So mineral fertilizers are actually only needed when microbes are absent, and no organic matter means no microbes, and therefore we can't feed the plants. Only microbes solubilize minerals for plants, so no one ever fertilizes forests because they are full of biota, full of life, doing the conversion from rock to soil to plant. Just to also show you this in another perspective, these are the major elements and minor and trace minerals, and these are the recommended ranges that we sort of work by in agricultural circles, and then the average in ag soil is already that much, and old growth forest has very minimal numbers. Tiny, the soil has almost nothing in it. This is why when we cut out the Amazon and then graze cattle on it for 2 years, it becomes completely depleted because there's no more plants and no more microbes that keep cycling those nutrients that were bound up in the forest. So old growth forest soil is useless without its biota, without its plants and organisms, especially the microbes, but also the other organisms that feed upon what the microbes extract. So we never ever need to fertilize these forests because they cycle those nutrients themselves continually, and the soil, it doesn't matter what's in it because there will be organisms that will get it as long as plants are present demanding those nutrients, and as long as the organisms are there to want those nutrients for themselves. So that's a huge take-home message. And of course, nutrient availabilities change as well. And when we test these, it's a very tricky situation because sometimes we just use different tests and they give us different results. That's why mineral testing of soils is kind of useless in the end. It might give us some indication of what's happening in the soil, but it's a very, very rudimentary test. And it depends which test you use that will give you different results. So really, it just doesn't really add up. But nonetheless, total soil nutrients, it's when the soil is ground up, digested with strong acids and combustion, and then it gives you the total amount. But then that is different from what's exchangeable nutrients, which are bound to organic matter and the sand and the silt and the clay and the organisms. So they're the ones that can do that exchange that we talked about earlier, where they can be moved in and out of soil solution depending on their absorption to different materials. Then there's soluble nutrients, which are actually dissolved in water and available to the plant. So that's the only thing that we're concerned with. So the best analysis really is to look at a plant tissue and have a look at what is actually within the plant itself. Has it absorbed all the nutrients that it requires? And what are the differences in certain way you grow the plant that will then show you whether those minerals are present? And if they're not present, we know that there's microbial deficiency where those nutrients are not being mobilized from the sand, silt, and organic matter and rock and whatever else, and are not getting into the plant. So there's a microbial deficiency, if anything. And this is in a diagram showing exactly that, where we have total nutrients, and that's minerals and organic complexes that are present that we can test for. Then the exchangeable nutrients that are attached to sand, silt, clay, organic matter, and the biology and inorganic compounds, and they can be, released. These pools can change from those nutrients depending on the microbes going into this exchangeable pool. And then there's also from the exchangeable pool potentially staying there or going into soluble plant available nutrient pool. And that's where this is interesting. And also that's where fertilizers are applied to increase that pool. But as I said, there's no need, because as long as you've got the microbes, if we return the microbes and allow them to do their job, they will keep getting soluble nutrients for the plant as the plant requires. There's no need to have it sitting in the soil. Just like the old growth forest has no nutrients present in the soil because it's all bound up in the life. And there's different processes that affect this, obviously, and that's chemical as well as biological. So this is the story of nutrient availability in soil. However, when we apply inorganic fertilizers, we must know that they destroy this process of the cycling through microbes to the plant. We're literally removing the microbes, and we become the supplier of those nutrients to the plant where the microbes would naturally do this. What the problem is with that is that all the fertilizers are salts, and so they dissolve in water, and that includes sulfates and nitrates and phosphates, and the problem is they attract water molecules to their charge. And you can see here we've got sodium chloride crystal as an example, and the water molecule and the sodium chloride molecule, once they dissolved in here, they're just sitting in solution. And now the hydrogen wants to bind with the negative chloride, and that means it just combines together, hydrogen chloride, and oxygen combines with sodium. And because that's negative, the oxygen and the sodium is positive. So they basically change, they swap over their partners that they were normally in a crystalline form sitting there doing their thing. And now that creates a salt solution. Basically, when you look at it on the scale of looking at what happens in the soil, any water molecule is taken away now to bind with this sodium chloride or whatever other salt we've added. And this basically takes water away from the creatures that are water-filled. It draws water out of them, it dries them, and from the plants. So we need to use more water in order to keep our plants happy enough to keep growing. So it's kind of a very, very wasteful, and it's very energy expensive to make inorganic fertilizers. And it's completely not necessary because for millions of years, nature has shown us that it grows giant forests without any fertilization. So we don't want to dehydrate soil and kill microbes. And when you actually look at it from a scientific perspective, this is shown clearly as well by Dr. David Johnson. I encourage you guys to look up his ways of dealing with farmers in teaching them how to create composts and really get the growth that the farmers desire from their plants, not based on nutrients, but based on the microbiology. And to show you this, his experiments showing that when nitrogen is in its peak, it has completely zero correlation with the plant biomass. In fact, plant biomass is highest when the soil nitrogen is at its lowest. There's completely no correlation between these two graphs. And the same with phosphorus, no correlation of growth of plant biomass versus a concentration of soil phosphorus, and the same with potassium, and even the same with organic matter. And what is found was the only correlation is between how much fungi and bacteria there are for a specific plant and whether that plant has got the right fungal to bacterial biomass ratio. And when it does, it correlates perfectly with its growth, and that's because all the minerals are now being supplied in the right amount to the plant as it needs them to grow. So it's the microbes that need to be present to get the minerals to the plant, and that's their job, and that correlates perfectly and statistically with plant growth. So plant growth biomass is directly correlated with fungal to bacterial biomass ratio, and that's something we'll cover obviously more in the future lessons. So we must remember then that sand, silt and clay is not soil, and it is the mineral matrix for organic matter and soil life. That's all these things are, and it's mineral matrix and food obviously because the minerals come from that matrix, so we definitely still need sand, silt and clay. But if the biology is missing, nutrients are not available to the plants. And the father of soil science, Hans Jenny, at the beginning of 1900s basically has stated that it was the sand, silt, clay and organic matter and aerobic organisms, which are key, that actually define soil. And anaerobic microbes can actually kill your plants. The reason for it is because anaerobic organisms can only live in anaerobic soil, and plants need oxygen in order to grow. So if organisms are present that are anaerobic, it indicates that your soil is suffering, but also the plants by default will be diseased because they are dying. So they need to be decomposed by the very organisms that we call pathogens. They're basically decomposers of these plants when the conditions are perfect for them to grow. So the agricultural industry tells us that it's sand, silt and clay and NPK, nitrogen, phosphorus and potassium, that are the definition of soil, but there's no mention of micronutrients. And also there's a complete dismissal of the fact that those minerals can only be added to the plant when the microbes are there to allow those to become bioavailable. And we should never give away all of our funds and everything to the chemical production and make us dependent on it when it's actually not what is needed and creates all these problems, waste of money, waste of energy, and kills biodiversity. So it's a living thing, and Soil is basically what is made by plants and associated microbes, solubilizing and cycling nutrients from sand, silt, clay, and organic matter. And you can see here that in a gram of healthy soil, there might be 1 billion bacterial cells. There would be 200 meters of fungal hyphae. The hyphae are just cells of fungi growing and extending throughout the soil in these networks. Sometimes we call this mycelium when it's visible in the soil. And also there are several thousand different taxa, different creatures that feed on these two groups or feed on each other, with the nematodes and protozoa feeding on these two groups of organisms to start with. So it is the soil food web that makes soil possible. And you can see here that in the micro scale, you have the little bacteria sitting on those, making those biofilms and communicating and extracting that mineral rock to start with. And then other organisms can gain from those minerals by consuming these bacteria. So they're the ones that are growing in these little nooks and crannies, protected in the soil aggregate, and that soil aggregate will also have oxygen present, and moisture can enter these gaps, and that makes a beautiful little system for the plant to access moisture, access nutrients directly from the bacteria that are being consumed on their roots, and access oxygen as well easily. And of course, with the fungi growing on their roots, the symbiotic partners, there's a huge amount of communication going on and hierarchy of how this process occurs in terms of what the plant needs for its best growth potential. So we have now come to the end of lesson 1.1, covering the mineral component of soils and how plant nutrients fit into that scenario. In the next lesson, we'll continue to look at soil formation with a focus on soil organic matter, soil ecology, and the soil food web organisms and how it all functions as an ecological unit. We'll look at soil structure, which is related to both soil organic matter and soil ecology. And then we'll look at ecological succession of how habitats are formed, how vegetation develops over time on a successional scale based on what is happening ecologically below ground. Okay, so you shall hear from me in lesson 1.2. Bye for now.